Yeah.
All right, let's go.
Okay, let's go. So Dr. Person is a professor of psychology and biophysics at the University of Colorado, and her lab studied cerebellar contributions to motor control, focusing on how the circuits as a whole generate behavioral relevant computations. She did her PhD at the University of Washington in the lab of David Perko. Then she did a postdoctoral studies at the National Institute of Biological Sciences in Beijing under Min Luo, and then a postdoctoral studies at Northwestern University under Indira Rahman. So with that, it's all yours.
All right. Well, thank you guys for having me. The Your group at Western is so wonderful. It's quite enviable, and it's also a delight to present this specific work because I feel like you guys have contributed so much to our understanding of short latency responses, long latency responses to perturbations. And so I have a lot to learn here and I appreciate any feedback. So as you requested, this is a bit of a journal club about this paper. The preprint is up here on bioRxiv. This is the thesis work of Courtney Dobrat, who recently defended her thesis, and so all of the credit goes to Courtney, and all of the blame goes to me. So, by way of introduction to the general questions that were what we're interested in the lab broadly, my lab really focuses on how the cerebellum contributes to motor control. And we've been long interested in this characteristic hallmark of cerebellar damage, which is unstable movement endpoints. And so this is demonstrated in this man who has a unilateral cerebellar infarct. I don't know if it's glitchy over there, but he touches his nose smoothly with his unaffected side here, but with his affected hand, there's this very prominent endpoint oscillation. Now, these oscillations have long interested cerebellologists because they resemble the kinds of oscillations that emerge in engineered control systems that use long sensory feedback or delayed sensory feedback to guide output of a controller. And so because these kinds of oscillations emerge with bad cerebelli, it suggests that the good cerebellum is solving some sort of a control systems problem. But the real question is what is that solution space? What is it actually solving? So one sort of class of possibilities is that it's generating some kind of a predictive control signal that mitigates the need for sensory feedback because it's operating in a learned predictive format. A mutually exclusive possibility, although an alternative possibility, is that cerebellar output is a sequence of small corrections that helps guide the movement to its appropriate trajectory or appropriate endpoint. And that it's generating these rapid corrections to structure the movement as it approaches its target. And so we've been interested in sort of dissecting the computations that the cerebellum builds in order to address some of these questions. The basic architecture of the cerebellum is extremely simple. It has a major input pathway through the mossy fibers that impinge on granule cells, which structure activity in Purkinje cells, which generate some kind of output. And we sort of assert in the lab very often that this output of the cerebellum is a predictive controller. And those of you who have seen me give a talk before may be familiar with this working hypothesis, which we kind of build out over several studies. The idea being that this mossy fiber afferent pathway has all this contextual information, efferents copies from motor cortex, sensory feedback signals from the periphery, potentially information about the motor goal. That information gets restructured by the massive input layer in the cerebellar granule cell layer. and generates a sensor motor or temporal basis set. This is actually real new recordings from granule cells during reaching with calcium imaging. So now we can even say that we think that there is a nice, beautiful temporal structure in the granule cell layer. So these granule cells then form these parallel fiber inputs to Purkinje cells. And there's a huge amount of evidence indicating that those the weights of these parallel fibers are under strong plastic control by error signaling or feedback signaling from a structure called the inferior olive. And so through reweighting of this temporal basis, we think that we can generate the population activity that we record in Purkinje cells that is structured around the kinematic representation or kinematic control of reach movements. And when we record in the cerebellar output nucleus, the interposed nucleus, we find many neurons that have activity that's scaled to the pauses that we see in Purkinje cells. And we know through closed-loop optogenetic experiments that that can actually structure movement kinematics. And so the idea then is that when this neural activity from the output of the interposed nucleus is too high, the animal will undershoot the limb. Or undershoot the target. And when it's too low, it may overshoot the target. And if it's just right, it appropriately structures the endpoint of the movement. So this activity we've always evidence suggests that this exerts an inward pull on the limb. And so we have, repeatedly concluded that we think that interpret this data as a predictive command of some sort. But it leaves open all this question about, well, what is going on with actual feedback? I mean, we know that there's sensory feedback coming into the cerebellum. So maybe this neural activity that we see is not predictive in any way. Maybe it is merely reactive. And that these reflect all these little corrections that are going on in the movement. And so that kind of ambiguity, I would say, in the current state of affairs with this data drives the question that we address in this paper, which is, what is going on? Are there reactive signals in cerebellar output neurons? And if so, how might they be structured by cerebellar output? Okay, does that all make sense? It was kind of a whirlwind intro. Okay, so to sort of frame what's known about corrective submovements, there's a huge literature on this, and these are two snapshot papers. But of course, Andrew is like the world expert on this, so I don't know if I have anything really to add here, but more to say that there is evidence that the motor cortex is intimately involved in generating corrective submovements. So this is a study from Teja Balu in Jesse Goldberg's lab, who used a joystick in mice where they're instructed or trained to push the joystick to a target region where they receive reward. And what they find here is that there are these little speed bumps in the trajectory or velocity of these joystick pushes, and that those are sensitive to inactivation of the caudal forelimb region of motor cortex. And so suggesting that there is cortical involvement in generating these corrective movements. This is consistent with a long list of papers, including those from Western, where motor cortical responses to perturbations include very rapid responses, and it is thought that those transcortical responses mediate some of these response to perturbation. Interestingly, though, the responses to perturbation are influenced in some ways by cerebellar inactivation. So in this study, there was a cooling probe inserted into the deep cerebellar nuclei that may include some of interposed. It's a little bit ambiguous and somewhat focused on dentate, but in any event, the observation was that the later components of the corrective movement in response to perturbation become a bit delayed with cerebellar cooling. And so while this takes cerebellum out of the immediate reactive phase to a perturbation, it allows cerebellum to have a sort of potential role in structuring the correction itself. And so in this example, the data are hidden by faces, but over here in this cooling phase, you can see that the responses become a little bit oscillatory in the absence of cerebellum or in the presence of its cooling. Okay, so what about unperturbed movements in mice? Like if we want to start to link neural activity that we've measured in interposed nucleus to corrective submovements, do we have anything to actually look at? So we use mice as our model organism, and mice are trainable to reach for targets. This is just an old example of a mouse reaching for a food pellet. It may be glitchy on your end, but in any event, these mice reach for food pellets, and we can use a variety of tracking methods to monitor the position of their limbs, both online and offline. And we can, offline in this case, analyze the reach kinematics and try to identify epochs where we would characterize some kind of trajectory deviation as a submovement. So in this case, we are defining these submovements as a accelerative deviation during the decelerative phase of the movement. So if the limb is already slowing down to target and it accelerates, we're calling that a corrective submovement. There's an additional definition that we have to invoke for these types of submovements in the early phase of the movement when it's actually accelerating. So in that case, we use peaks in positive jerk because that indicates an increase in acceleration during the acceleration phase. So these kinds of sort of classifications look like this, where these sort of zero crossings in positive acceleration are what we tag as the time of a corrective, or actually in this case, so far, I'm just showing you submovement. We can analyze the distribution of the timing of these submovements, and this will become relevant later on. But we notice this very characteristic peakiness to the distribution of these submovements. They cluster in these little clumps that have about a 65 millisecond delay between them. We don't know what the significance of that is, but it is this characteristic distribution. And that helps us sort of characterize when these submovements are actually occurring. So we can quantify when these submovements are happening. I would say half submovements of these submovements happen in the early phase of the reach, and then the rest are distributed throughout the sort of decelerative phase. Most reaches have them, but not all of them do. So that's nice because it allows us to compare and contrast reaches that have an overt submovement and those that don't. So the first question is, do these submovements actually mitigate error in any way? We've looked at this in a couple of ways. And so this plot simply illustrates a submovement. So this is the zero crossing in acceleration. we can see that there is a deviation, of course, between reaches that do not have this submovement and those that do in the kinematics. And this is also true in the kind of velocity view of this, where this reach that eventually has a corrective submovement is clearly different from a typical velocity profile of a normal reach. And that sort of uptick in velocity or increase in acceleration sort of gets that reach back on track. So we can look at this in these z-score plots, which I think are a little bit dizzying, but let me just walk you through this. So here we have each sort of epoch within the reach that can have a submovement. And then we look at the deviation of the kinematics prior to the time of the CSM. Recall that in these T early, we're using this jerk definition, and so our acceleration is a little delayed from that alignment point. But the point here is that you can see that there's a clear deviation from a normal reach in this indicated in this dark purple, and similarly in the velocity, and that becomes mitigated. So it's much lighter after the CSM, and then this is the CSM that's defined in this dark green. Probably more to the point, when these corrective submovements appear to stabilize endpoint position. So despite having these corrective submovements, all reaches and all types end up with a common or non-significantly different endpoint. So these kinds of data argue to us that these submovements are corrective in some way. And so then that allows us to ask this question about what their representation might be in neural activity within the interposed nucleus. So here we're using acute neuropixels recordings. These probes are lowered into the anterior interposed nucleus. These mice, of course, are now head fixed. So this is a slightly different setup than the previous recordings from INT-A that we have presented previously. But similarly, we see neurons that have concerted activity right around the reach epoch. And you'll have noted probably that we separate these neurons into these groupings of early and late phase activity. So this is just a heat map showing all of our INT-A neurons. You can see that there's this kind of, again, this plurality of neurons with peak activity right around during the reach. We can kind of zoom in on that region and look at these heat maps of peak activity around peak velocity. And what you can note, I think, easily is that there's a real distribution right across that phase. So there's some that are active during the acceleration and some active during the deceleration, and it's just a sort of stretch along that temporal axis. These are stably represented, however. So if we separate out units by even and odd, we note that the early units are stably active in the early phase, whereas the late units are stably active in the late phase. And so this suggests this sort of continuous tiling of the reach and potentially continuous control. We can get into that if you really, really want to. We think that based on some subsequent analyses that I'm not going to show you right now, that these late units are correlated best with the decelerative phase of the movement, and these early units are correlated with acceleration. And so linking this with some optotagging and I guess, transsectional optogenetic activation experiments that Ayman Azim's lab has done, we think that this may represent two different populations of cells that either push the limb out or pull it back. Okay, so that's kind of our set of populations. So then the question is, well, what are they doing during these corrective submovements? So here we're looking at some late units that are aligned to around the midpoint of the reach. And we've separated these out into reaches that did not have a CSM and those that did. And I think you can probably largely appreciate that the difference in neural activity is extremely subtle here, if anything. And so we would argue that this kind of data suggests that it doesn't appear to be the case that the major signal coming out of the interposed nucleus is associated with this kind of corrective movement. On the other hand, we did see a subset of units that did have distinct neural activity when there was versus when there was not a corrective submovement. So we can, what's going to happen here? Okay, so this is the actual figure. So we can subdivide these out into what we're calling a tuned population versus an untuned population. And I guess the key thing to note here is that in this tuned population of late neurons, these cells appear to be active during the late phase of the corrective submovement. So this is the delta of the kinematics between a reach without a CSM and that with. And you can see, so this is the deviation associated with the corrective submovement, and this is the peak in the neural activity in the tuned population aligned to that time point. So sort of going back to the observation of Willis and Hoare, we would say that this is not driving a submovement, but it may be involved in structuring it kinematically in some way. So if we interpret these particular cells as pulling on the limb, then they may be shortening or adjusting this CSM to sort of bring it to an end in a way that's pretty analogous to what we think that same population is doing to structure the deceleration of the reach to begin with. Okay, so this sort of gives us an insight into these spontaneous corrective submovements, but these are tiny and they are and they're not induced. So the question becomes, well, what happens if we induce a perturbation to the limb, and does it correct? And if so, are there neural correlates to that correction? So the experiments that we perform in this study take advantage of a couple of features of cerebellar output neurons that we think are premotor. So first of all, the structure itself contains both excitatory and inhibitory populations. These excitatory populations project to many downstream areas, including the motor thalamus, the red nucleus, and other parts of the medulla. And there are there appear to be subpopulations that have distinct projection patterns, but the red nucleus is a massive target for these cells, and we wanted to distinguish perturbations here from potential effects of activating inhibitory neurons as well. So we used an intersectional approach where we inject a channelrhodopsin into the interposed nucleus this is the actual experiment. So we inject a Cre-on and flip dependent channelrhodopsin into the interposed nucleus, and we have previously injected a AAV retro that expresses flip only in int neurons that actually project to the red nucleus. And so we're doing this in a VGLUT2 Cree mouse. And so only excitatory neurons that project to the red nucleus are going to be labeled. And that's what this is. And we're going to contrast that to our previous experimental paradigm where we inject all of the interposed neurons with a non-Cree dependent channel rhodopsin. We then perform a closed loop optogenetic perturbation of reaches. Here we're doing this on 25% of reaches. This is a very brief 50 millisecond perturbation that is triggered around the midpoint of the reach on 25% of trials. And this is the same perturbation type that we've used in a previous publication. So we'll sort of get into why that becomes sort of interesting. Okay, so this is what this kind of experiment looks like. This is actually the INTE-ALL experiment, but the duration of that perturbation is very brief, and it interferes with the reach trajectory. So these data are those we've already presented many, many times. This is the typical reach velocity when there is no stimulation. And when there is a stimulation, there's a very rapid decrease in outward velocity. When we restrict the channel rhodopsin to the RN projecting excitatory neurons, we see that these kinematic effects are almost identical across the two types of perturbation. What deviates, however, is the response to the offset of that stimulation. So when the train of light stimuli is over, we see no concerted or in the average kinematics, we don't see any kind of initiation of the reach in these INTE-ALL stimulation conditions. But with the INTE-RN, there's a very prominent reacceleration of the limb, and this ends up correcting the movement endpoint entirely. This is the same data, but just in acceleration, and you can see that right towards the end of that stimulus, there's this massive acceleration that occurs, and it's poorly timed, and in the average doesn't appear to actually correct any movement in the INTE-ALL condition. So the question is are these real corrections? And so one argument is that if they are corrections, they may scale to error magnitude. And so here we've plotted the, this is the sort of acceleration of the corrective submovement versus the effect of the stimulation. This is for the INTE-ALL condition, and this is for the INTE-RN condition. And I think you can easily appreciate that in the INTE-RN condition, across all mice, the magnitude of the correction scales better with the magnitude of the perturbation, such that it's closer to the line of unity than it is with the INTE-ALL condition. And that has an effect then on the actual endpoints of these movements. So as we've emphasized many times over the years, when we stimulate INTE-ALL, when we stimulate all the cells in the interposed nucleus, we cause hypometric endpoints. And this is even despite the fact that there are sometimes corrective movements. So there are reinitiated reaches in that condition, but they end hypometric. So there's this misscaling of that corrective movement. Whereas with these cell type specific perturbations, we find that the endpoints, even though we're causing this massive deviation in the limb velocity, they come right back and they're they're right there with them, and there's no significant effect on the success rate. Okay, so then we can start to ask questions about the effect of experiencing these corrective submovements on how the typical reach is actually structured. So one of the first things we looked at was, of course, the timing of these corrective submovements. So this is the timing of the corrective submovement that is in response to the induced perturbation. So we stimulate, the animal generates a corrective submovement. What we noticed is that, so I want to remind you that the distribution of spontaneous corrective submovements had this sort of interesting peakiness to it, and it was sort of distributed throughout the reach duration there. We noticed that the timing of spontaneous corrective submovements changed drastically after exposure to the perturbations. So these are spontaneous CSMs, and after the animal has experienced this closed loop optogenetic perturbation, the timing of these spontaneous corrections shifts a whole bunch. And so we found that kind of intriguing because it suggested some kind of a relationship between an experienced perturbation and when a correction might happen. Now, it's an important point that although in the preprint we sort of emphasize that these become predictive, they're not happening actually before the stimulus-induced perturbation itself. So please just view that with a bit of an edit there. But nevertheless, there's this large distribution or shift in the timing of these corrective submovements in the spontaneous condition. Now, with the inte-all condition, so this is when these corrective submovements are happening in response to the stimulation, you can see that they are sort of widely distributed compared to that very sharp peak in response to the stimulation of the inte-RN population. And then when we compare the spontaneous events after exposure to stimulation, we find that there's not a development of a learned peak there. And so there's room for speculation about why this might be. And there's, like any bad experiment, there's multiple variables here. The variable that we're quite interested in is the fact that when we're stimulating inte-all, we are stimulating an inhibitory population of neurons that project to the inferior olive. And so we speculate, but we're not sure, that activation of those cells may interfere with the learning of this timing. But of course, the experience of the timing of the CSMs is also different and may influence the what is learned as well. Let's see, neural correlation is blocked. Let's see, I have to press OK. Okay, so then of course the question is are there neural correlates interposed in response or associated with these corrective submovements that are induced by the perturbation? And so this is these are the kinematics that I'm showing you again. So here's this large decrease in the velocity in response to stimulation. And here are some unit responses. So as we would expect, we see many units that have a very strong reaction to the channel rhodopsin stimulation. And then during the corrective submovement, we see this big increase in neural activity in these cells. So once again, this activity seems associated with the correction, but it's not obvious that it is either driving the correction or really like, predictive of it in some way. So we think it is reflecting, we think it's associated, we think it's probably shortening the perturbation, but it's less clear that it is really doing the driving of it. This is just the same data except separated out into early units and late units. And again, so this dash is the time of the CSM, and the early units are active a little bit before that, but it's not as if their peak is well before the timing of that CSM. And similarly, the late units are delayed. And so it's kind of interesting to us that the timing distribution of these units that we see during the typical reaches is maintained during the corrective submovements themselves, as if they sort of recapitulate in some way that patterning during the entire movement. Okay, so I gave you a little bit of a hint that the experience of these perturbations influences the structure of future unperturbed reaches, suggesting some format of learning, some evidence of learning in the system. That shift in CSM distribution happens in the first session. And it becomes difficult to study the evolution of that because it's so fast to emerge. However, we thought, well, we could potentially ask a question about learning in response to cerebellar output stimulation that addresses a couple of open questions. So one sort of piece of background information is that we know from work from my lab that we can perturb reach kinematics by optogenetically activating Mossy fiber inputs to the cerebellum. But very interestingly, if we stimulate again and again and again, the magnitude of that stimulation effect goes away, or it diminishes. So in this example, our unperturbed reach is in gray, but upon stimulation of the Mossy fibers, we induce in this example an overreach. Okay, so that's the perturbation, that's the perturbed reach kinematics. But after we stimulate, not too many times, in fact, just probably about 10 trials, the magnitude of that effect is already diminished. And this is despite the fact that we're still stimulating. We went on to show in this paper that if we take the stimulation away, we see an after effect, etc. And so the question is do we see a similar profile of adaptation when we're stimulating cerebellar output instead of cerebellar input? The reason this question is interesting is that, at least as far as this goes, this kind of adaptation could be happening outside the cerebellum. There's no law that this had to be within the cerebellum. We argue that it was because we saw neural adaptation there. But it is entirely possible that this entire learning profile was inherited through changes from other regions of the brain. And so the question then is, if we do the same block structure of stimulating cerebellar output, what do we see? So the first thing to note is that if we separate trials where we've just begun the stimulus in a block structure, we're calling this stim early, versus those that are late in the block, stim late. If you look, you cannot even distinguish the kinematic effect early and late in the session. So the magnitude and scaling of these stimulus effects are identical early and late in training. So that's quite distinct from what we see in the with the Mossy fiber block stimulation, and suggests that it's not obvious that anything outside of the cerebellum can, say, ignore or turn down the effect of cerebellar manipulations. So kind of just to draw the comparison here, the effective stimulation early and late to cerebellar output does not adapt, whereas it does when we're doing it with the input. So then what's going on neurally? So this gets into the final figure of the preprint, where we see that although the effective stimulation is absolutely unchanged during the stimulus block, early and late, when we remove the perturbation, we see a subtle increase in reach velocity and an faster progression of the reach itself. And so this suggests that there is an after effect, even though there's no overt adaptation during stimulation. And we think that that's important because it suggests that there could be a site of learning in the cerebellar cortex, probably in the Purkinje cells. And what we're doing when we stimulate the cerebellar nuclei is blocking the expression of that learning, but not the learning per se. So then if we sort of pan over here to the neural activity, again, this is the after effect that's in pink. Here's the neural data. These are the firing rates during the stimulation in blue, and then during washout in pink. And you can see that there's this very prominent and I think extremely intriguing dip in int A firing that's associated with the timing of that optogenetic stimulation. It's beautifully matched, and so and that's quantified here. So we are very intrigued that this is a learned signal and a signature of adaptation going on in the Purkinje cells that is driving this this dip in firing that is countering the opposite direction effect induced by the optogenetic stimulation. So to kind of tie it all up, the question was kind of how these sorts of reactions and corrections are incorporated into cerebellar motor control. We found that there do seem to be some subtle neural correlates of these spontaneous corrective movements. And then we can drive these large corrections in response to perturbation that shift the timing of spontaneous corrective movements, and that over multiple stimuli, that there is adaptation of the neural activity that appears well-timed to counter the opposite direction optogenetic perturbation. So sort of pulling it all back to this larger picture of what we think cerebellum might be doing, we think that it's likely that there is some structured recurrence here, and that the feedback either through motor cortex from the periphery or even sort of broad outcome signals get broadcast back to the cerebellum and can be reincorporated into the patterns of activity that they use with their little knobs to control components of reach velocity through their sort of privileged connection to specific muscle groups through the limb. So that's kind of where we're at. There's a number of sort of open patient components of all this, most notably relating these early and late cells to these subgroups that have recently been identified by Ayman Azim's group. And yeah, maybe we can just stop there, and I'm happy to do my best with answering questions. And I'm, yeah.
Thank you.
Yeah.
Yeah. Hi. Thank you for the talk. Great, great. So I wanted to ask, what do you think is the role of vision in these corrective movements? In other words, what do you think would happen if you occluded vision from the mouse while doing the task? It seems
Yeah.
slightly different to me from the patient going like touching their nose, because like you know where your nose is, right? You don't necessarily need visual motor control, whereas when you are reaching for a target you might need um, more visual feedback.
Yeah, I mean, I think that the intuition that we build as highly visual primates is not right for these mice. I don't have the data to show you this, but I do know that in other experiments from our lab that these mice will reach very well in the dark. So they are not. This is not a highly visual task. They are locating these pellets through, in the head fix condition, much more through whisking. And Dan Huber's lab has shown that olfaction is a huge component of the sort of targeting of these movements. And so I do not think that these corrective submovements are visually driven. That's my speculation. And moreover, mice are not terribly visual creatures. So does that maybe that's what I think. So I do not think that these are visually driven, and we would not make a claim that they are. Yeah.
That might explain also the distribution of those corrections, right? You said they are mostly at the beginning of the movement rather than later on, like
Yeah,
when.
there's a majority, but then they have this kind of cool little bumpy distribution. Yeah.
Yeah. Thanks. Thanks.
Yeah. Um, Interestingly, though, like this kind of non-uniform distribution is seen in humans as well.
Yeah, but in humans, I would expect it to be more towards the end of the. I would expect more corrective movement towards the end of the reaching in humans.
Yeah, but they're. I guess my point is that they're not uniformly distributed.
Yeah, no, that's
Yeah.
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 Okay.
Yeah.
Great talk. So I was like, for your first result, I kind of stopped listening there. No, I didn't stop listening there, but that
Tell
was
us what you really think, Jorn.
no, but the so you stimulate the red nucleus projecting interpositus neurons, and you get the same deceleration, but then the system issues these submovements, and in a controlled experiment, when you stimulate the anti-interpositus, then you shut down, you decelerate the movement, and you kill the subcorrective. So
Yeah.
have you done the experiments where, I mean, okay, so now the question is the pathway of decelerating the movement and somehow totally disrupting the system so it can't even do a correction after the stimulation is gone? Is this different pathways? Meaning, like, have you done trying to block the thalamic projecting interpositus neurons or the reticular ones?
So
And just
we don't know what's issuing this. That's a good question, but I do want to emphasize that these red nucleus projecting neurons are the same neurons that project to the thalamus. They collateralize. So almost all of these output pathways collateralize widely, and it makes answering your question actually extremely difficult
okay. So when
because
you compare the two stimulation conditions, which
right.
which group of neurons do you not stimulate when you do the reticular, the red nucleus ones?
Yeah, so okay. So the groups of neurons that we do not stimulate are we do not stimulate the inhibitory neurons that project to the IO. That's kind of my favorite population. We do not stimulate, there's a very small subset of neurons that project directly to the spinal cord and do not go to the red nucleus. So we don't stimulate those. That's an interesting population for our purposes here. But then also, I think that once we have all of this recurrence, what is the motor cortex informed about? Like that's a wide open question. So it would be very interesting to know what motor cortex knows under these two different regimes.
You mean, okay, so I mean, you haven't done the experiment from trying to get the inhibitory neurons in the interpositus and project to the IO. Okay, is that technically possible?
It is, and yeah, yeah. I think the reason that I don't know about this is that it's not obvious. So what we're left with is being able to turn off the IO neurons to ask whether they're, sorry, not the IO neurons, the nucleolivary. So those inhibitory neurons are called the nucleolivary neurons. We can manipulate those and we can look at spontaneous CSMs. But as I mentioned, I think that those spontaneous CSMs I don't necessarily think that they're driven by cerebellum. And so the question is whether they're influencing the response to the induced perturbation. And that's a really hard experiment because of the way we're inducing the perturbation, we're optogenetically activating the other cells, and then we'd have to optogenetically inactivate those other inhibitory neurons. So in some ways the int-al condition is that, but it's an imperfect experiment. I'm sort of in sort of thinking about this revision, pretty sure that we might not like emphasize this because we do not have a good answer to it. It was just an interesting observation that popped up in comparing and contrasting these effects, but it's a difficult thing to chase down. But I mean, I really do feel like it's got some qualities to it that could sort of motivate a bunch of future research, because it's cool to be able to absolutely knock out the corrective submovement.
Well, if you did the experiment with stimulating the inhibitory neurons in the IO, in the interpositus that goes to the IO,
yeah,
then you get the deceleration in the movement, first of all.
What? No, you don't.
You don't, you know that.
Yeah, we know that.
Okay. And therefore, then you also don't get increased submovement.
That's right.
So that's why you talk about spontaneous. Okay, that's interesting. Okay, so I didn't know that.
Yeah.
Okay. So then, this difference between these two conditions could be either a mass effect, right? When you stimulate all the neurons, you just like wreak more damage on them. You perturb the motor system more, so the mouse is just giving up on that reach. That's one thing. And if you only do the N and thalamic neurons, then the perturbation is somewhat smaller. But that doesn't seem to ring right because your deceleration is matched, right? So
yeah, we definitely perturb it in both ways. Yeah.
Yeah.
Now here's another speculation. What do you think about this? You guys are the experts here. So, if with the int-al condition, let's say that we're activating a larger population of the excitatory population that includes those that go down directly to the cord, and it includes this population that's going through brainstem and up to motor cortex. And maybe one thing that's different is the actual reflex. So I think that one component of the actual response to stimulation probably, and I mean like behavioral response, it must include spinal reflex. And so if there's a contribution of cerebellum to spinal reflex, and this has been kind of long speculated, is that there is some sort of gamma contribution to gamma motor neuron activity, that if we interfere with even the detection of that error, then we would disrupt the generation of a CSM, and then all bets are off because the timing is screwed up, and there's nothing to learn. So that's kind of another direction. So I've kind of written a grant about all these speculative ideas, but so I don't know. I feel like they fall into different classes. One is that IO, this population to IO is critical. Two is that the reflexes themselves are disrupted, and I guess those are kind of two key ideas. Yeah.
Can I ask a question about the nature of the perturbation itself? I mean, it seems to me like you're making your life quite difficult because you're studying the thing you want to see the corrections with and inducing the errors, inducing the thing by perturbing those neurons, basically. And I'm wondering why you wouldn't put yourself in a little bit of give yourself an experimental handle by, for example, moving the food pellet mid-region, inducing a corrective movement. Now you know that unless you think that's something categorically different that you're not interested in studying, if you think there's something special about, I don't know, you know what I mean? So you can induce it experimentally. Now you know it's going to happen. You have some idea it's going to happen. And now you could interfere and wiggle around these different circuits and see which one changes it, how much does it change it, and so on. So I'm just curious about that version of it. Maybe that simplifies your life and gets you dissociation, like handle experimentally on the problem.
Yeah, I mean, it's a good point, and I think that there's kind of a lot to unpack here. So on the one hand, sure, we could do response to target perturbation. That is different than an actual perturbation to the plant or the brain, right? And so it may or may not induce the same neural responses, but certainly we could study it. We have designed a target jump that will jump in closed loop. I think that the more sort of analogous perturbation to all the stuff that's been done in humans is actual, like a perturbation to a joystick.
That's another
And huh?
Yeah, that's another version. Yeah, yeah.
Yeah, right. And so and absolutely. I mean, I think that those are very interesting questions. And so if I had actually gone into this with like, oh, this is really what we're going to study, that might be what to do. We had all this data on these responses and decided to chase them down. So we noticed.
That wasn't
Yeah.
meant to criticism or anything. I was just wondering
No,
logic.
no. But I think
Parsing those out would be potentially incisive for this to answer this question, I guess.
yeah, I mean, I think that I don't know how incisive it is because I feel like each one of these approaches asks a slightly sort of orthogonal question, and we might only get a full picture with all of them together.
But let me just ask,
Yeah.
like, if you
Yeah.
have a nice model now, if you are, let's take your loading example of the limb. So you have some scheme, which I don't know how you build it in a mouse, but for us it would be easy. We would put the animal in a robot and the animal would reach, and then some moment we would make it, we'd increase the viscous load or something, and now the animal, in order to overcome that and still get the reward, is gonna have to jack up the muscle activity associated with reaching out. Now, in the cortex, in the cerebellum here, and these various kinds of neurons we can't do this part, you could do this part with labeled neurons and understand precisely what kind, which projection class, and when they're activated in response to generating that correction, right? And then you can manipulate the size of the load, you can there's a lot, then of course your imagination runs wild after that, right? And I don't.
Sure. Well, I mean, but we do know some. I mean, we know some of this from monkeys. So,
Well, none of this beautiful stuff with the cell type, the projections, all of that is not.
none of that. Yeah. Sure. Yeah. No. I mean, everything you said is right, and I think this is the beginning of sort of building that out. And yeah, for sure.
Thanks, Abby. I have a quick question about the timing of the simulation. So
Yeah.
would you get the same response if you just change the timing of the opto simulation to very early or very late in the reach? Or this is like this golden time point that it only affects in that time point?
So we know that we've only done the timed experiment with the inte all condition. In that case, we always see the same kinematic effect. And in fact, that was the one of the critical arguments that what we're doing is pulling on the limb, because if we stimulate during return, we pull the limb back faster. So we're not just seizing the limb. We did not do that with the inte RN condition. So I can't tell you, but
Thank you. I was
I,
also wondering if you had a chance
yeah.
to look at the population level, like because you have a lot of neurons now, but looking at the like a status space of all of them and seeing like, is there like a specific direction in that status space that is changed by the stimulation or not, rather than just looking at single cells or the average response in a sense.
Yeah, I mean the question's a good one. So we haven't done a state space analysis on these data per se. All of the sort of state space trajectory analyses we've done of Purkinje and nuclear populations have, I mean, it's been a little bit puzzling what they tell us. We know that there's a bit of a sort of sharper kink in the state space trajectory with inte neurons compared to Purkinje neurons. We think that it's just reflecting the fact that that's kind of how they're rather sparsely activated during, or their bursts are kind of short during these experiments, and so they don't overlap a great deal. But if you have ideas about what we could pull out of those state space trajectories, I'd love to hear them.
The one thing I was just constantly thinking that if you train like a linear readout of that status space for the position of the arm essentially during that one dimensional reaching, then you can see how much of this perturbation that you induce in the neural state actually projects to that potent movement inducing direction and how much of it is outside of that. Since especially you see also that learning effect, you might actually see kind of be able to connect how this like gradual multiple stimulations are kind of shifting you in a dimension that are not in the output, but can be used for the learning part of the data.
It's an interesting idea. I mean, I'll keep thinking about it. Certainly, the neural adaptation that we, or that dip that we see that sort of corresponds with the timing of the optostim. It's easily interpretable in the potent direction of what we think that neural population does, which pulls on the limb, and then we're doing the opposite, which is freeing up that. And so, but I do agree that there's probably some deeper analyses we could do there to sort of get at the relationships across the population. I mean, this is something I've been really fascinated in is, you think about these feedback loops. To what extent is the sort of these late phase signals sort of baked into the cake from the beginning, or are they sort of in constant communication with each other? So yeah, I am intrigued by some possibilities there, but it's not immediately obvious what we're going to pull out. So
Yeah.
I like your idea. Yeah, thanks.
How directional are these? I'm just trying to like, I'm thinking that, I've read Iman's paper and I guess you have, and there he has these two directional populations. So he wonders, but I'm curious about whether or not you can get the mouse to reach in different directions, for example, and whether these undershoots are really reflecting undershoots per se, or whether they're reflecting kind of more, whatever the muscle group population would be that's relevant for outward movement.
I'm not catching the distinction. I mean, I think that we're yeah.
If the mouse reached towards its body instead of away from its body towards a pellet somehow, I don't know how you do this experiment. and now you stimulated these exact same populations of neurons, would you get undershoot? Or would
No,
you,
no. So this is why we talk about it as a pull. So we've done the experiment when we stimulate when the limb is returning to the body.
okay, you have done that, okay.
Yeah. And in that case, the limb gets pulled back faster. So it's an inward pull.
It's an inward pull, that's what I
Yeah.
wanted to know, okay.
Yeah.
Very interesting. Very interesting. Okay, any other questions for Abby?
Thanks, Abby.
Good. Thank
Yeah,
you
that was
so much.
amazing.
Thank
Thank
you.
you.
Hey guys, thank you so much. I appreciate the feedback, and if you have thoughts or if you have other lingering questions, please don't hesitate to reach out. This is kind of a new direction, and we're, as you can see, kind of building it up. So you guys are the experts, and it's great to
you.
hear from you. So okay,
Have a great day.
Thank you. Have a great day.
bye bye.
That act of-
Yeah. We don't